Yeah, thank you for this nice opportunity. So I'm not a professor yet. I'm still a postdoc come researcher. Uh, my research is motivated by the problem of understanding nanoscale processes which consume energy and perform systematic work outputs. So these are called non-equilibrium nanoscale processes. And according to me, there is no better place to look for these processes than the most basic unit of life, which is a cell itself. And in my research, I use machine learning as a tool to understand questions which otherwise are very hard to answer. So that's going to be the theme of my talk today. I'm going to tell you how I'm using machine learning to answer certain questions which would be otherwise very challenging. So to start with, let me start with examples of three cellular processes. So on the left side, you see the process of cell division. This is the most fundamental process in biology. And here what you see is a cell, when it is dividing, is separating out its genetic material, which is chromosomes shown in pink color, into the two daughter cells. And in the center, what you see is a cell called fibroblast. It's part of the connective tissue in our body. And in this particular experiment, they have put the fibroblast in a isotropic circular confinement. Yet, despite that, when it is developing, you see that it is sort of developing these patterns which break symmetry. And then, there you see a chiral symmetry breaking and the swirling towards a preferential left direction emerging. And in the third case, if this movie works, it's actually not working. So here again, I have a fibroblast. What if the movie was working? You would have seen it moving in a extracellular matrix, the cell itself navigating through the extracellular environment. And so now these processes, cells execute them with high precision and balance in fluctuating environments. Once in a while, this precision is lost. So, for example, if the cell division doesn't occur correctly and the genome is not equally separated into the daughter cells, then you have cancerous conditions. So, one of these conditions is called aneuploidy, when certain daughter cell gets more genetic material as compared to the other. Or if a cell is moving around much more than it's supposed to, that also leads to spreading of cancer, and so on. Now, these processes are not free. They are driven by what are called molecular motors, which are nanoscale microscopic engines that derive energy from the environment, often by breaking simple sugar molecules and then performing useful work. Most of this captured energy from the food that we eat is used for fueling directed cellular processes such as cell division or cell development. But a large amount of it is also dissipated as heat to the surroundings. Now, that is the key quantity that I'm interested in my talk. If you know the heat dissipated in a particular process, then you have an idea about how much energy is being used up. So there is this question about how much energy is a cell spending and when is it spending more when it is dividing. And if you also know what is normal levels of dissipation, then that also gives you an idea about conditions such as cancer or neurodegenerative diseases where there can be more dissipation. So nowadays we can visualize and probe these cellular processes in very good detail. Like these movies, you can easily, or with certain expertise, you can make in our labs. And we also have a good understanding of the detailed biochemistry of these processes. By which what I mean is we know what proteins and what chemical reactions are involved in cell division or cell development or cell motility. But despite this, I believe there are two critical open problems. The first problem is what I refer to as the inference problem. It is the problem that for cellular processes such as mitosis or cell division, we don't know the exact local energetic cause. Local in the sense of local in space and time. When a chromosome is being pulled out and separated out into two daughter cells, we don't know which stage of this process is the cell using most of its energy. Now, why is this so hard? It's hard because the energetics involved at the scale of a cell, which is 1,000th of the thickness of our hair, is of the order of few kBt. 1 kBt is 25 millielectron volt, and that is much below the detection threshold of standard calorimetric techniques. So we cannot put a calorimeter and measure some energetics that happens when a cell is dividing. And our best estimates otherwise come from complex chemical reaction pathways. You have to kind of keep track of what chemical reaction is happening, and then how much is the energetics associated, how much is the binding energy released, for instance. But this is going to be too complex, even for a process like cell division, because it involves hundreds of proteins and hundreds of chemical reaction pathways. That is the first problem. Now, the second problem is what I call the control or design problem. This is basically the fact that cells, as I show, or living systems, as I showed you in the previous slide, are able to assemble biomaterials with targeted properties, and they do that systematically over and over many times. But in contrast, if you were to reconstitute all the things that would have made that beautiful swirling pattern, we wouldn't be able to recreate it the way that a cell does systematically. The main reason is that the physical mechanisms and driving forces behind many of these cellular processes are unknown to us. And we also have very limited control in these active and fluctuating environments. So these are the two questions that I seek to answer. And then I want to show you how you can make headways using results from non-equilibrium statistical physics, which I will introduce first, and then combining that with tools from machine learning. So first we focus on this inference problem. So in the inference problem, the key quantity is energetics of processes at the cellular scale. And pretty recently, over the last 25 years, there has been significant progress in the statistical physics and thermodynamics of such processes. And this field is commonly referred to as stochastic thermodynamics. And the main ideas of this field is to look at energetics and dissipation and its fluctuations and so on of non-equilibrium processes at nanoscale. And the basic ideas can be introduced using this very simple system. It's a system of a colloidal particle. That's a micron-sized particle which is not miscible with water. It is big enough that you can see, but it is small enough that it will experience thermal fluctuations from the environment. Such a particle, you take it in an aqueous solution, and then when you put when you apply a laser beam, you can basically focus it, keep it focused at its focal point. Now, in this experiment, the size of the particle is something like 3 micrometer, and the force that the particle will experience is of the order of piconewtons, and the distances that it moves is of the order of nanometer. And it turns out that these systems have energetics of the order of cellular processes that we are interested in. Now, let's say we have this system, and now let's say I do an experiment where I am moving so I'm holding a particle at a position, and I'm moving it to the right. And I'm also changing the power of the laser beam. Maybe I'm holding it weaker and weaker as I'm moving it to the right. So this is my first experiment. Let me call it the forward experiment. And then I do the reverse thing. I bring back this particle from the final position back to the initial position. So I do 2 different experiments. One is taking a particle from left to right, and then I bring it back from right to left. Now, if you look at the trajectories of these particles, they won't be a straight line deterministic trajectories. It's primarily because of the effect of the bath molecules. They collide with this micron-sized particles, and as a result, the particle fluctuates quite a lot. And so you see these stochastic trajectories. Now, if I were to repeat this experiment, I wouldn't be starting the particle from the same position every time because of noise again. And I wouldn't be having the same noise history in every realization. So as a result, if I repeat the forward or reverse experiments many times, you will get very different realizations. Now, this is dynamics. What stochastic thermodynamics gives you is a framework to compute how much work is that one is doing when you are taking a particle from one position to the other. And then it also gives you a formula to compute how much heat is dissipated when you bring back this particle from or in the same experiment or in the reverse experiment. Now it turns out that, as I said, if you again do this experiment many times, you'll get different work values for different experiments. So you get probability distributions of work values. it turns out that there is this nice result that the work distributions of the forward and reverse distributions in this particular case look something like this, and there is a point where they overlap. And the point at which they overlap is related to the free energy difference between the initial configuration of the particle and the final configuration of the particle. Now, this has been called, this is called fluctuation relation, and it's been proved in generality for Markovian processes. And interestingly, now, I mean, it looks like a nice mathematical result which applies to a cute experimental system, but it's actually of very much practical applicability. Now, let's say you consider this other experimental system where I have a DNA hairpin and I have put a bead at one end of the hairpin. I put it, kept it there, and then I'm pulling the other end of this DNA. So this is one end of the DNA where I have attached a microbead and I kept it fixed at a position, and I'm trying to open up this DNA hairpin by pulling this other end here, which is shown with red, because that is the optical tweezer that you use. And if you do this experiment many, many times, you'll get similar work distributions for opening up the DNA hairpin and closing the DNA hairpin. And of course, these distributions will overlap. As you see here, this blue one here is a work distribution corresponding to the filled circles that corresponds to the stretching experiment where you're opening up the DNA hairpin, and the not filled, the sort of unfilled circles correspond to the one where you're closing it. So from these points where they overlap, interestingly, you can find out how much energy is released when a single base pair is broken. And what is the binding energy released when you break a single DNA bond? And that value comes out to be about 5 times 10 to the power minus 20 joule. And this is, again, along the lines of what I said before, it's such a tiny quantity of energy which you wouldn't be able to measure using any calorimetric methods. But stochastic thermodynamics gives you a way of computing energetics at such tiny scales. Now, in my PhD research, I was looking at a large class of analytically solvable systems like colloidal particles in traps. And then I was trying to look at work distributions of these systems analytically. And then we looked at a bunch of cases, which included, for example, particles in a trap whose mean position is modulated by a noise which is called active. And then, for example, particles having viscous and viscoelastic interactions, which are very typical of a cellular condex. And then particles experiencing multiple temperatures along orthogonal directions, so that they start going around the mean position in a systematic manner as a rotary motor, and so on. So we looked at a bunch of analytically solvable cases. But this is not the problem that I want to talk to you about today. All these systems are interesting because they are analytically solvable. They give you an insight about how you can develop methods in non-equilibrium statistical physics to compute dissipation and universal properties of systems out of equilibrium. But can they give you an idea about real biological systems, right? That is the question that I want to address. The challenge is, so you want to go beyond analytically tractable models. So the colloidal system that I showed you is an analytically tractable model. I want to go beyond that. And the challenge is only very few systems have, completely non-dynamical descriptions, an equation describing their motion, basically. And for example, for cellular processes like cell division, we have a lot of experimental data nowadays. But despite this, precise estimates of spatiotemporal forces or dissipative cause are largely unknown. So that is the inference problem. Can we develop model-independent ways to directly infer the energy dissipation and driving forces from experimental data? Now, personally speaking, I got attracted to this question by 2 papers which were presented at Nordita at different points of time. The first one is this paper where they look at this algae called Chlamydomonas. And what you see here is the protrusion of the algae from its head. So or what you call the head. Algae uses, it's a flagella, and algae normally uses this protrusion for locomotion to, sort of move around in the surroundings. and it also uses it for sensing the surroundings. Now, what they did in this experiment is they reconstituted this, hairy protrusion, and then so you basically just take out the hairy protrusion, and then you put it in a medium which provides the energy for the motion to happen. So it is an ATP rich medium. ATP is the currency for this process. So basically, the flagella is reconstituted in a ATP rich medium, and then you see that it starts going around in this systematic manner in a circular motion. And what they then did is you take the state of the you take the state of this flagella at each point in time and then decompose it into the first three Fourier modes. One could go beyond this. Let's say we just try to find out how much of each Fourier mode is contributing to the flagella shape at each point in time. And then if you carefully bin sort of the statistics of this dynamics, in the sense of you try to look at the transitions between these three states now, then you will see that there is a nice flux in the phase space. So such phase space fluxes would not have happened if the system was, not doing this. Like if it was just randomly fluctuating, or if the algae was dead, you wouldn't see this systematic motion. So these currents in phase space indicates that there is a dissipative dynamics. Now, is there a way to go from this dissipative dynamics to dissipation? Like, can you quantify how much energy is being used up when a flagella is moving around in the systematic fashion? So within stochastic thermodynamics, I won't go into the details. There is this idea of local detail balance. It basically says that if you see certain transitions happening in your system, it is being in contact with the thermal reservoir. So there must have been an energy exchange between the thermal reservoir. And then sort of extending the ideas from equilibrium statistical physics, you say that the forward to reverse rates of transition would have given the ratio of the rates of transition in the forward and the reverse between, let's say, this first two states would have given you the heat dissipated in that particular process. Now, this is an hypothesis, and this hypothesis correctly gives you the equilibrium statistics when the process is actually in equilibrium. But so in principle, one could go around, so looking at these states, so take your data, decompose it into different possible states, and then you can look at the transition rates between different pairs of states, apply this hypothesis of local detail balance, and then that would give you an idea about how much energy is being spent in that particular process. So this is possible in principle, but in practice, as you see, it's going to be very challenging. I mean, if I have not three, but let's say four states, already it's challenging to visualize that. I mean, one cannot really visualize fluxes in 4 dimensions. You'll have to cut it into different sub-dimensions. Also, in statistical terms, binning the high dimensional data becomes more and more challenging as the dimensionality increases. That's the curse of dimensionality. Okay, so but nevertheless, people have applied these methods in a bunch of analytically solvable systems and shown that you can, in principle, get estimates of dissipation using this kind of brute force binning technique. Now, the other result that I got attracted to at the same time is called as the thermodynamic uncertainty relation. Again, I'm not going to the actual details of the derivation of the result, but what the result says is that, so if you measure currents of the type, so these fluxes that you see are currents in the phase space, and if you do measure this for individual realizations, they will look different. Or if you measure it for a certain duration of time and then measure it again for the next 10 seconds, that will be different. There will be fluctuations in the values that you of actual currents measured. So if you measure the fluctuations of these currents of the type that you saw in the previous slide, and in particular, if you construct a quantity which is like a Fano factor, it's called estimate of precision of these currents, that gives you a lower bound to dissipation. And this was again proved for a broad class of systems which obey a Markovian dynamics. It was shown that if you measure currents in the phase space and then you measure their fluctuations in a particular way, that will give you a lower bound to dissipation. And I'm denoting dissipation rate by sigma, that is the rate with which energy is dissipated in this particular process. And here tau is the time duration of the process, kb is the Boltzmann constant, and again J is the particular current that I'm measuring. So this was shown. This was shown in 2015, but there were it was interesting because you don't need the details of the dynamics. You only need experimental data from a biological process to be able to measure a lower bound to dissipation. So at least that at least the amount of energy spent is this much by just measuring experimental data. However, there were two crucial disadvantages. First one is that the estimated lower bound can be arbitrarily low. You don't have any control over how low the value that you're estimating is. I mean, you will get a value very close to zero, but the actual dissipation could be way much higher than that, and there is no control over how low this lower bound is. And then the second one is the only known case where the bound saturates was the equilibrium limit. So in this particular figure here, you have a two temperature system studied, and dissipation in a two temperature system studied as a function of the ratio of temperatures. So when the ratio of the temperatures are 1, then you just have one temperature and the system would be in equilibrium with that temperature. When the temperature ratio is different, then the system is further away from equilibrium. And here the red line you see is the true entropy production rate or true dissipation rate in the system. And the black and blue lines are what you estimate using the thermodynamic uncertainty relation. As you see, the values agree when the value itself is zero. As in, the best estimate you get is when the system is in equilibrium or when the system is not dissipating. So that's not really an interesting scenario for an estimator. So naturally, this sort of got a lot of attention in the literature, that thermodynamic uncertainty relation is a nice result, but it has this limitation that it is not saturating in any non-trivial case. So in my research, while doing a PhD, we were able to show we were interested in studying processes as a function of time, and we studied a bunch of analytically solvable models as a function of time. And here you have a particular case where the actual dissipation in the system is 2. So 2 is the value that the system is actually dissipating. And with different lines here, what you see is the dissipation estimated by different currents in the system. And I have also plotted it as a function of time. And as you see here, if you were working at a large time or a finite time, typically the value that is estimated is much below the true value. So the true value is 2, and all of these currents are estimating something around 1.5. But then we studied this as a function of time. And if you go shorter and shorter in time, you see that there is a lot of variability, but there is in particular one current which is saturating the bond. That is, the thermodynamic uncertainty relation saturates in the short time limit. So if you measure shorter and shorter currents in your system, then it is bound to saturate the thermodynamic uncertainty relation for a broad range of problems. And here, not only that, the current that is saturating the thermodynamic uncertainty relation is what we call the dissipative force field. So if this force times the displacement of the system would give you the total entropy production or total dissipation along that particular trajectory. So by identifying the current saturating the thermodynamic uncertainty relation, you will also have an idea about what is the dissipative force field sort of running the system. And our proof was based on fluctuation theorems and generic arguments. I'm not getting into the details of that. There were also alternate proofs provided later on. So now the interesting aspect is we have a methodology where you can measure different currents, and then in principle, one of those currents would saturate the thermodynamic uncertainty relation in the short time limit. And then that could be used as an estimator, then in practice for dissipation. But how do you implement this in practice? So this is the first instance where I acquainted myself with sort of machine learning. And so we do the following. Let's say I have an arbitrary system whose phase space degrees of freedom is denoted by xt. I measure a long trajectory in the system. The way then one proceeds is that you chop it into very short trajectories because you want to now measure currents in the short time limit. So you chop it into many short trajectories. And then if you know the thermodynamic force field, then you can work with these short trajectories and compute dissipation directly. But you don't know that in practice. You only have the trajectories available to you. So what we do is we model this thermodynamic force field using a neural network. And the architecture of the neural network and what features that the structure of the neural network should have, etc., should be crucially dependent on the actual, if you know some symmetries of your problem, then you could make a smarter guess for the neural network. And in the future, we are trying to work in that direction. But right now, we are working with very simple feed forward, type neural networks. And then you perform a gradient ascent of this cost function, which is this thermodynamic uncertainty relation given cost function, where at each step in your optimization, you update your force field, which is now a neural network, trying to maximize this value. So in particular, if you are able to measure all the degrees of freedom in your system, and if you are able to work with get short enough trajectories, then theoretically this procedure is bound to give you an exact estimate of dissipation. And this, since you can work with neural networks and there are very efficient algorithms to perform optimization in machine learning, the procedure is scalable to higher dimensions, and this people have already shown. So now what we did is we tried to benchmark this in a bunch of experimental systems. I'll show you results from one of these studies. So the system we considered is particle in a harmonic trap whose mean position is modulated by an Ornstein-Uhlenbeck process. So you have 2 degrees of freedom here. One is the position of the particle itself, and the second one is the mean position of the trap. So here x is the position of the particle, lambda is the mean position of the trap. Okay, that movie is not really playing, but you would see the particle moving and the trap also moving when the experiment is being carried out. So here we have a bunch of straight lines corresponding to different, so the blue one is the theoretically predicted value of dissipation, and there are a bunch of error bars. And you see that for a range of values of theta here corresponds to the amplitude of shaking the particle, we get estimates of dissipation within reasonable agreement of the theoretically predicted values. Not only that, this method also reproduces the dissipative force fields pretty well. So here on the left-hand side, you see the dissipative force field that is inferred from data using this machine learning algorithm. And on the right-hand side, you see the dissipative force field that is predicted by theory. And they agree very well to nanometer resolution, of course, and piconewton values. And not only that, you can now take the knowledge of this force field that you learned from the data, put it back on the trajectory, and then identify where and when is dissipation happening. So I can now color the trajectory that you see on the left-hand side by the value of dissipation at that point. And then you can see you can color the same trajectory with dissipation and tell which parts, so in this particular phase space, where is the dissipation higher? Sorry for this poor color bar. This green here means low dissipation or zero dissipation, essentially. So at the center of the trap, the particle is not dissipating, but then everywhere else, there is asymmetric type dissipation. This you can now measure, and everything is just from the experimental data. So one doesn't use any knowledge of the experimental system. We measure the trajectories, pass it through this machine learning inference algorithm, and it tells you from just dynamical trajectories, you get this information about how much dissipation, which is the thermodynamic quantity, is happening, and where is it happening, and when is it happening. And that is a very nice result, I think. And of course, one would get the same thing if you use the knowledge of the dynamical equations that is producing these trajectories. Now, the real interesting question is, of course, one more thing is that we can also apply this method to time-dependently driven systems. So if I take a particle, like the experiment that I mentioned at the beginning, I'm moving the particle or I'm changing the power of the laser that is holding the particle in the center. We can make many copies of this experiment and then look at a particular time slice, a window, and then apply this algorithm. And then you can then get a time-dependently driven estimate of dissipation purely in a data-driven manner without using any knowledge of the underlying dynamics or the experimental parameters used. Okay, cell membrane is one of the most dynamic and integral part of a cell. It plays a crucial role in signal transduction, cell-to-cell signaling, cellular uptake, and much more. And the interesting part is the role of various biomolecules and on the structure and dynamics of the cell membrane can be systematically analyzed. We have a good knowledge of the underlying biochemistry of a cell membrane. So we did this experiment where they looked at the height for, so here what you see is a HeLa cell. It's a lineage of a cervical cancer cell. And what you see in this movie is the height fluctuations of the membrane of the cell. So the flickering that you see is the change in the height fluctuations of the membrane of the cell. And here, then we take this movie, and then there is a conversion factor that you have to convert the light intensity into the height of the membrane at that point. Now, we can also look at this in space and time. We can look at it across the membrane of surface of the cell membrane. You can also look at it in time, in region, locally. And then from that data, so now you have a time series of height fluctuations across the cell surface and in time. Now we take this data and then apply this algorithm, machine learning algorithm on this data. So now so here is how the schematic, how it looks like. You have here, what you see in the dark gray color is the cell. That's the background where the cell is kept. And you take a region of, let's say we take this blue patch here. And for each such patch, you have a movie. You have a movie of how the height is varying over space and time. We take that data, I pass it through the inference algorithm. First, do a dimensional reduction to sort of capture the most dissipative, most dynamic components. And then you apply it through an inference algorithm. And now each point in each region in this patch, you just had the height values. Now that once you pass it through this algorithm, you can convert that to the dissipation in that region. And we did then a systematic experiment where we depleted the cell of ATP. So you have a healthy HeLa cell, and then you treat it with a drug so that it is essentially killing the cell. So now the cell has much less access to ATP than before. So at time t equal to 0, you have a healthy HeLa cell. And then at 20 minutes and 40 minutes and 60 minutes, it's essentially dying. Now what you're measuring is the dissipation from the cell using this machine learning algorithm. And as you see, there are 2-3 observations that one can make. First of all, the cell lights up when you apply this algorithm. So more red color means more dissipation, and more blue means basically zero dissipation. And the cell region is now well distinguished from the background. And then within the cell itself, there are regions, there is high inhomogeneity. There are some regions which dissipate more. There are certain other regions which dissipate much less. And then the third thing is that when you deplete the cell of ATP, the value of dissipation goes down and the surface color reduces. And so when the cell is healthy, it dissipates more. And when it is not healthy, it dissipates much less. Or when it is killed, it dissipates much less. And this is sort of the first direct measurement of dissipation in the sense of stochastic thermodynamics. And then you cannot directly put a calorimeter for a cell membrane to get this locally in space and time like we can do with this data. There was also another experiment done where we treated the cell with a drug which weakens its cytoskeleton, and you will see a similar effect that the dissipation goes down when the cytoskeleton of the cell is weakened. So to summarize this part, this method sort of spatiotemporally resolves energy dissipation and microscopic forces from experimental data. And it's applicable to time-lapse movies of fluorescent labeled processes or just natural contrast movies. You don't have to put a tag, or you don't have to really experimentally probe the system. You can just let the cell do what it is doing and make a movie of that. It's still minimally invasive, but very minimally invasive. And then use that data and then convert that to an estimate of dissipation. And of course, there are many systematic lower bounds imposed by the experiment because you're only able to see the membrane fluctuations. You don't see anything else that is happening underneath. So your best estimate just comes from that time series that you have access to. But still, it gives you a very good estimate of dissipation. And the process, since this procedure, we are able to use neural networks, we can easily scale it to higher dimensions. And as I discussed, we have shown that this method works for, at least theoretically, for complex and systems having complex energy landscapes. And even if there is an explicit time-dependent driving, then also you can use this method to measure dissipation. So I think I'm almost past or like closing 30, 35 minutes. How much more time do I have?
Unfortunately, we are not in a program, and the time is over. And
yeah.
Do you have some more slides?
I just wanted to show you one more thing, but I can also stop.
Sreekanth, unfortunately, I think we have to stop. Just to have several questions, probably from participants of the seminar, and after that we should move further. So please, questions. Probably people are thinking about questions, and I would like to ask about experiment you show in some slide of unzipping of DNA hairpin. You used this in this experiment. You use pulling with constant velocity or constant force.
So here the variable.
Yes, that's right. This.
In this particular, so this is experiment that was done. I was not involved in this experiment. It's a classic experiment that was done, 2009, I believe. 2009. And so you could choose the protocols. In the end, when they computed the work statistics, they always sort of stopped between two extension values. So you can basically measure the separation between this bead and this bead. And then when they computed these work statistics, they started from some initial value, which is presumably corresponding to a folded configuration. And then they unfold it for certain duration and stop it to certain length. And then, basically take measurements for that separation. So you start from a short separation and go up till a final separation, which is large enough where you have good confidence that it is unfolded. And that's how they did it. But then, you can have constant force protocols or constant velocity protocols. And I think there is a lot of flexibility, but the cells generically hold.
Okay, but I see this is the work 2008. Yes. Okay. Thank you. Other questions? Yes, Professor Bridget.
Sreekanth, thank you very much for interesting talk. Could you please comment on the time dependence of the thermodynamic uncertainty principle?
Yeah.
This is time of the process which you study, or please?
Yeah, so there are two aspects. So here, originally when the the thermodynamic uncertainty relation was conjectured, it was shown that your process is in a steady state. So there is a stationary dynamics. And now you can measure currents for certain duration of time. Current is for example, the heat dissipated itself is a current, and that you can measure for certain duration of time in a steady state. So that is one time dependence coming, how long you keep on measuring these currents and how long. And so there is, even if the system is in a steady state where there is a time translation invariance, you can choose to measure currents for a certain time duration. You can choose to take data for a certain time duration. And that has an effect on the uncertainty relation. And that's what we showed initially, that when you take currents, measure only very short currents. Don't keep your measurements for very long time, but you just work with very, very short currents. Then the uncertainty relation itself improves and saturates, basically, that we showed. So that is one aspect of time dependence. The other aspect is that the process is not a stationary state. There is an explicit time dependent driving, like in this particular case, you have a particle which is, who's trapped with a laser, and its power is changing in time. So initially the particle is confined in a very small region. Here it's position versus time, and then it spreads out. And there is an explicit time dependence. So dissipation itself is now time dependent. And in this particular case, what we showed is that if you choose a time window, then time is a parameter. I mean, at that particular window, you can treat essentially time as a parameter and think that there is a equivalence in this ensemble across different realizations. And we showed that the method applies here and then gives you an exact estimate of time-dependent dissipation, using the same procedure. So there is the actual time dependence of the process, and then you have a process in a stationary state where you measure, for different time durations. So these two variables come in TUR. Was it the question that you asked?
Okay. Sreekanth, thank you very much for a very interesting lecture, and we hope that we will have opportunity to listen other material that you wanted to tell us, but unfortunately we do not have time.
Yeah, no problem.
Okay, thank you. Let's thank Sreekanth.
Thank you.
Thank you very much. And we go to other lecture. And the following lecture is by Remi Monasson from École Normale Supérieure. And the lecture is When Statistical Physics Meets Machine Learning, 40 Years of Boltzmann Machine. So, Professor Monasson, you can share the screen.
Yes, can you hear me, actually?
Yes, we can hear you and we can see you.
That's great. Okay. So thank you so much. So I would like to first thank the organizers of this very kind invitation. So I was a bit surprised because I'm neither Swedish nor Ukrainian, but I will try to be up to the task. Okay. So, in fact, as was said just before the first talk by the organizers, this year's Nobel Prize in Physics was a bit special because it was, let's say, at the crossroad between physics and machine learning. And my talk is going to be concentrated on that. And I would like to go back to Boltzmann machines. And Boltzmann machines were actually the center, let's say, the focus of one half of the prize to Geoffrey Hinton. And I would like just to start with a quotation from the Nobel Academy. So Geoffrey Hinton used tools from statistical physics to create the Boltzmann machine, which can learn to recognize characteristic elements in a set of data. The invention became significant, for example, for classifying and creating images. That's a quotation I took from the website when I prepared this presentation. So what I would like to do here in this talk is actually to go back to what Boltzmann machines are, because maybe some of you are not familiar with these machines, since they're a little bit on the borderline of physics, and try to look at, let's say, modern applications of these machines and also interesting theoretical questions we are still open. And I would like to tell you about some recent works in this field. Okay, so let me start with Boltzmann machine. And I think if we want to understand what Boltzmann machines are, we have to do one step backward, which is going back to the Ising model. So the Ising model is something we teach all students in statistical physics. It's really a cornerstone of undergraduate studies in statistical physics. And we all know what it is. Essentially, you have a model here in a two-dimensional grid, but it could be any kind of geometry, three-dimensional, for instance. And we have a local magnetic moment, which may be up or down. So it is strongly anisotropic material. So let's say +1 or -1, depending on the direction. And an energy here, which is written on the right, where the Jij are usually equal to J, which is positive or negative if you have nearest neighbors on the grid, or 0 otherwise. And there have been a lot of work to understand the nature of the phase diagram of this model. And essentially, given the model, we want to compute observables like the average magnetization, spin-spin correlation, and many other quantities of interest. So the problem that actually Hinton and collaborators looked at is the inverse problem. So can we, from data, guess what the Jij are, and can we build a model which is actually focused, adapted, adequate for a particular set of data? So let me try to explain in a better way what that means. So suppose, look on the left, suppose you have some data, and I've chosen here some MNIST digits, which are just part of a famous data set, quite old actually, in data science, which are just handwritten digits that were used by the US postal system. And each digit here is a 28 by 28 grid image. And each pixel is a + or -1, depending on whether it's black or white here. So suppose you have many of those digits. Actually, the actual database, I think, contains about 60,000 digits. So what we would like to learn is an Ising model that is an Ising energy function of that kind, where the Jij will be fit to the particular data set in such a way that if I plot the energy as I'm doing here on the right side, here are the VIs, which are the different configurations of the pixels. So 28 by 28, this is 784. So there are 2 to the 784 possible configurations. Then we would like the minima of this energy function to coincide more or less with the data items here in the database. And we would like maybe the top of this energy function to correspond to things which are essentially random, actually that do not look like at all digits, right? There should be some kind of blurry images that we don't care. So if we are able to do that, then that means we have learned an energy-based model, which is actually adequate and focusing on our data. And that's the core principle of Boltzmann machine. So the question is, how can we actually learn this energy function and the Jij parameters from the data? So there are many questions which are interesting. As I said, the learning, how does that work? Then if you have learned this energy function, how can you sample it? So, and then there are many interesting questions because, as I said, those minima may correspond to the digits here in the database, but there might be other minima. For instance, that one here, I hope you can see my mouse. That one was not a data item, but maybe it's a nice looking digit. It's something interesting. So we can use this model to generate new data. Then there are also questions about the interpretation. What are these Jij parameters which are learned from the data? Do they have any specific meaning? And then there are also interesting questions about low energy excitation. If we have an energy, it's nice to try to locate the ground states, but how do we move from one ground state to another one, to a next one, by flipping maybe some kind of small collective modes, I mean, limited collective modes, groups of spins to go from one state to the other? Can we understand the low temperature physics of this model? So let's look at the first question, which is how do we learn the model from the data? So let me define the problem. So we have some variable VIM. So I runs from 1 to n, which is the dimension of the data point. So for the MNIST digit, this is 784, as I said before. And m will be the number of data points. So capital M, so small m will be the index of the data points. So here in the MNIST databases, this is capital M is 60,000. So these are the data we are looking at. And what we would like to find is a probability distribution that corresponds to an Ising model. So with some Jij, which are unknown, maybe some local fields, which I call GI here, which are also unknown. And then this Gibbs distribution here with the Z, which is the normalization, the usual partition function, is defining the probability of a configuration over capital N spins. Right? And it depends, of course, of the unknown parameters J and G. Okay, so how do we learn the J and the G from the data? When what we do is we do some kind of maximum likelihood inference, as people call that in machine learning, which simply means that we would like to maximize the product of the probabilities of the different data items, or if you take the log, the sum of the all data items of the log of the probability which I have written here. Okay, and the data are here, but of course, this thing here is just one big function of the different unknown parameters, the Gij and the Gi, and that I will call minus an effective energy. So it's an effective energy. We have to be careful, not in the spin space as we are used to in statistical physics. It's an effective energy in the parameter space, in the model parameter space, right? So it's a very high dimensional space. And we would like to minimize this effective energy V. And what the Boltzmann learning machine algorithm, which was introduced by Hinton and collaborators in 1985, is simply a gradient descent of this energy here. So what you do, you imagine some kind of fake dynamical scheme where you have this learning time t, which is not a physical time, right? It's a time on the computer, which is just, so we have gradient equation. We just go down the gradient of V along the G parameters, the local fields, and the local interactions here, Gij. I mean, the interaction, sorry. And if you compute explicitly this gradient, then there is a nice interpretation. Actually, the gradient with respect to one parameter, for instance, the Gij, is going to be the difference between the correlation function between Vi, Vj computed with the model, so with that distribution, and the value of this correlation in the data. So essentially, when you are reached the minimum of this function, V, of the effective energy, that means the moments in the data and in the model match, and they are called moment matching condition. So there are very interesting features about this learning dynamics. You can show that the solution is unique because V is actually a convex function of its parameters. So it's very nice. You are guaranteed that you will converge to, if you go slow enough and you don't do overshooting, and when you are close to the bottom, then you will eventually converge, find a nice solution. And in fact, there is also deep connection with the maximum entropy principle, which was also at the basis of statistical mechanics according to Jane's formulation. Okay, so that's how it works. So there have been a lot of applications of these techniques over the past 40 years, and I want to mention only one of them, which because I think it's a beautiful one, I like it, and it's quite recent. It was done over the last 15 years, and let me just mention it very briefly. So a very important problem in proteins, and in fact that was also another Nobel Prize this year, which was given to people who have actually worked out AlphaFold, which is a protein folding algorithm, which is able to produce a prediction for a structure given a sequence of amino acids, is to understand the relation between a sequence of amino acids in a protein and its function or structure. So here is a very simple domain with only 30 amino acids, which is able to fold, and its structure gives it some specific property, that is some specific capability of binding a set of ligands here, small peptides, right? And if you make some mutation along this sequence here, then the structure may be affected or not, and that could change the function of this protein. So the mutation can be neutral. Sometimes they don't have any effects. Sometimes they completely disrupt the structure and the function, and sometimes it may be actually beneficial. They actually increase, for instance, the affinity of the protein. So how this is connected to Boltzmann machine? Well, I mean, we need data to understand this mapping, and for instance, and so the data that people have been using, and here I'm citing some of the people who have been collaborating to, contributing a lot to this particular field, and Eric was one of them. So what we can use evolutionary related sequence across many organisms. So if you look at one particular protein, for instance, a polymerase, so all living organisms need to copy their DNA, right? To replicate their DNA. So all living organisms have polymerases, but they have evolved from the last common ancestors billions of years ago in a very diverse way. So we have a lot of sequences for proteins that essentially do the same task and have very similar structures. So we can use those data, and then we can infer a Boltzmann machine. We get an energy function. This energy function, this landscape I was showing you before, right? We get this thing here where the bottom here of the wells correspond to the good proteins, the one in the database. But then if we have this energy function, we can ask many things. So for instance, what is the impact of a mutation? Because if I do a mutation, I will go up or down a little bit along my energy profile. So it will give a score actually to this mutation, whether it will be deleterious or beneficial. And there are also very nice things about the interpretability of the model. The Gij, which are inferred in this particular case, they were shown to be actually related to the contacts on the three-dimensional structure of a protein. So it's a beautiful example of the applicability of a Boltzmann machine, and it was at the core of the first version of AlphaFold. Okay, so far so good. Many applications, I just mentioned one of them, but there is still a very big problem, which actually was already recognized by Hinton and collaborators 40 years ago in a 1985 paper. The problem is actually Boltzmann machine, they don't answer one of the questions I asked before a few slides ago. What about lower energy modes? So the question is the following. If I give you the set of parameters Gij, Gi, and that's actually what you get at the end of the inference procedure, what do you understand about the energy function? Of course, you are able to compute for any configuration the value of E, which is very nice. But do you understand where the minima are without doing the full exploration of the space? Very tough. Do we understand how two minima, which are next to each other, are related to each other? How I flip maybe a group of spins to move from one minimum to the other? No, we don't, right? In fact, it's a well-known problem in statistical physics. People have been studying spin glasses for decades, and that was the Nobel Prize 3 years ago, where we are given the Gij, we are given the Gi, and yet it's extremely difficult to understand what are the properties of the equilibrium measure in this landscape, right? So having the parameter is a constructing way to get E, but it doesn't mean that the physics is obvious to understand once we have them. So can we have a better way to represent the same energy function, which actually uncovers what are the nice, important physical features? Okay, and to do that, we have to change our perspective. We have to go from one way of thinking about data, which is the Boltzmann machine on the left here. Again, we have some variables, V1, V2, Vn, and there might be correlations between these different variables in the data, and we try to account for those correlations, finding some effective interaction. That's what Boltzmann machines are about, right? But we have this problem that we don't really understand how the energy function is made. And then the other interpretation, the other way of thinking about the same data is to try to understand the correlation not in terms of effective interaction between the variables, but kind of latent factor of variability between the different configurations. So the idea is to go from this architecture on the left to the architecture on the right, which is called restricted Boltzmann machine, which actually is incorporating a completely new idea from a physical point of view, which is the idea of representation. So essentially, what we want to find from a data configuration is a representation of the data, which tells us what kind of factors are active or silent in this particular configuration. So I'm going to make this more explicit in the following. But this latent factor, which explains the kind of correlated changes from one configuration to another one in the data, have to be found from the data, right? They are not given to us. We have data, so we know configuration on this blue layer here. We don't know anything about the green one. So we have to understand that. So this has to be extracted from the data. So how does that work? Well, I mean, it's an extension of Boltzmann machine. So it works exactly in the same way, except that we don't have only configurations from the data, but we have also these hidden configurations, which are called latent configurations. So now we have a joint energy, which couples the visible variables where the data are, V1 up to Vn, to new one, H1 to Hm, which are these hidden units or latent units. So we have terms which are local terms acting on the visible units. We have local terms which are more complex. I'm going to explain that, acting on these hidden units. And we have also couplings in between the two layers. So we have no couplings within the layers. We have couplings only between the layers. So that means that any kind of correlation between the V1 and V2 within the data have to be explained through interaction with H. So the H's will focus on creating these effective correlations, right? Okay, so how do we learn the model? Well, formally, if we integrate out these latent factors of variability, these H's, we define the marginal distribution over the visible layer, then we get an effective energy, and that's exactly, we are back to the Boltzmann machine in a way. So we want to maximize over the data set the product of the P of these on the data items we have. So an interesting point I want to mention is that due to the bipartite nature of this interaction graph, we may actually go both ways. Given a V, we can extract latent factor variation using the conditional probability of H given B. And if we have done that, we can go the other way. So that means we can generate new data given a representation using the P of V condition to H. And I will do both in the following if I have enough time, which I hope I will have, right? Okay, so what about the connection between this architecture and that one? I told you you can go from that one to this one. It's a new way of thinking about it. But in fact, the left architecture is a specific case. It's a special case of the right architecture. And I want to show that. So it's we just have to do a couple of Gaussian integration to show that. So it's an easy calculation. Suppose I choose U of H. So this U of H, this local potential here, as a square over 2. Okay, so a quadratic potential. It's a specific case. I can choose that. In that case, the effective energy acting on the visible and hidden units is that one. I just replace U of H by H squared over 2. Then I'm going to compute the marginal distribution. This is just the integral over H of the effect of exponential minus E, so the Boltzmann weight. And there is a normalization factor here. And that defines the effective energy. So, but now look at the H mu. They are all uncorrelated from each other. No interaction between the H mu if you take the exponential minus E. So, and they are just Gaussian variables. So you can compute this Gaussian integral, multidimensional Gaussian integral very easily because they are all independent. And if you do that, you get an effective energy whose expression is written on the last line here. This term obviously is not affected because it did not depend on the H. And we get simply the square of this term when we do the Gaussian integration. And if I exchange the order of a sums of the mu and I get a double sum over I and j, vi vj, with an effective interaction matrix jij, which is the sum of a mu, wi mu, wj mu, where these w's are the weights connecting the visible and hidden units. So you see, the Boltzmann machine is a special case of this more complex architecture, okay? And in fact, if I go beyond H squared, if I choose another potential, I will have another model, which is not a Boltzmann machine. So the RBM includes the Boltzmann machine as a specific case. So what are the interesting potentials we can look at? Well, it depends on what you want to do. So let me just describe. So the black potential, H squared here, is confining this later factor. So what does it do? Essentially, it prevents the H's to be very large, right? Because the potential grows very quickly when H is large. And in fact, that having not so much large H is important because it means that if you look at the conditional probability of V given H, if the H are super large, that means that the V's are highly constrained. So you have very low variability when you sample from that distribution. So if you want to have kind of expressive power in terms of the V's and not only a single ground state configuration, you don't want the H's to be super large, right? So it's super important to have a confining potential here. But you can do much more than that. You can also choose the V. So look at the blue one here, which has a non-zero slope here in 0. Okay, so a cusp, while the black one was flat, right? H squared. So why is it interesting to have a cusp? Because it means that you have a low cost for H equal to 0. And as soon as you move a little bit away, you will pay a lot, right? You will pay a lot. So it favors H equal to 0, which is something which is akin to L1 regularization in machine learning. So it favors sparse representation. You will have a hidden layer where many H's will be 0. That's nice because if they are 0, that means you have some kind of representation which contains only a few H's not different from 0. So it gives you interpretability about what is actually important in your visible configuration, right, in your data. So it's nice to have sparse representation of data. So you may have also, for instance, this red potential, which has a kind of bimodal potential, 2 wells. So the H's will be either that value or dot value, and it favors discrete representation, so clustering of the data in a high dimensional space of representation, right? So it helps separate and divide, partition the representation space. So how do we choose between all this potential? We don't have to choose actually. We just have to parameterize this potential and learn the parameters from the data, okay? So I just add the weights, they are also learned from the data. So we get nice adequate representation that fit as well as possible the data you look at. So let me show you one example. Sorry, it's going a bit fast. So here, this is a list again. I've already shown that to you. So we can learn an RBM using gradient descent of the log probability of the data. And then once you have done that, you can sample it. I'm going to explain how to sample it, but first I want to show the video. So you will see 10 different Monte Carlo chains. I'm trying to get it. Okay. So they're just independent. They are drawn from the same machine. Okay, so look at the top, let's say, look at the second one. So you see the machine is generating data here, and what you see is only the visible layer. We don't look here at the hidden layer, right? So the machine is running, okay? So it was trained with 400 hidden units, okay? And they correspond to this kind of family of potential I've shown before. And depending on the hidden units, the parameters are different. So you see, the only difference between these 10 different Monte Carlo chains is the initial condition. And then, of course, they are noisy, so the noise is not the same during the dynamics. But they correspond to 10 times the same machine. Okay, so what you see is that first, there is a large diversity. Quite nice. They look like handwritten digits, not every time, but most of the time, right? And I can assure you that if you look at them and you compare to the database, they are not in the database. It's very unlikely.
Mhm.
Okay, so that means actually the machine has a large entropy.
Excuse me, unfortunately, we have problems with electricity for this moment, and we just can't hear you. We do not see your presentation at the moment.
But you can hear me?
Yes, but we can hear you.
You are missing the best part. It was a Monte Carlo simulation. But anyway, I can talk.
Okay.
So essentially, the machine is generating data which are not in the data set. It's not simply a memory of the data set and which is, recording data, right? It wouldn't be so interesting. They are new. That means the machine has actually learned from a few data items, a complex distribution with a large entropy, and is able to sample from it. Okay? Please let me know when you think if you have back electricity and you can see the slide.
We need some time because we can't.
Okay, no problem.
Your slides. Just a moment.
Yeah. So I try to move on without the slide. Oh, okay. So now in the last part of the talk, I would like to go to two kind of modern extension. So everything I've said there was kind of from the 80s. That's actually not true because there were no very efficient learning algorithm up to the beginning of the 2000. And in fact, they were developed to do pre-training of deep networks because the Boltzmann machine, these restricted Boltzmann machine were very influential in the 2000, let's say up to 2010 to train these networks. But I'm going to that later. So the first question I would like to mention is how to do sampling of these machines. So the bipartite nature of the interaction graph gives you actually a very simple algorithm. You start from a visible configuration, which is, for instance, one data point that you have in your data set. Since there is no interaction between the hidden units, that means that condition to a visible configuration, all hidden units are independent from each other. So they are extremely easy to sample. You just sample real numbers. Very easy to do. Once you have your latent configuration, hidden configuration, you can sample back one visible configuration. And again, due to the bipartite nature of the graph, all visible units are independent from each other, very simple to sample. And you just repeat this process back and forth, okay? That's called alternate deep sampling. And in fact, the pictures I showed you, I hope you could see some of them. I mean, and the video I showed you was obtained this way, right? Once a machine is learned, you can sample it very easily. But there are some particular kind of data set where actually the sampling is very bad. And in fact, if I train MNIST only with 0 and 1 digits, so I just forget about the 2, the 3, the 4, up to 9, we can learn a machine, okay, which works well in the sense that if I initiate I start with an initial configuration, which is a 0 digits, it will sample a lot of nice looking zeros, but I will never see a 1. And if shall I continue? Is it okay? Okay, and if I start with a 1, I will sample many nice looking ones, but I will wait forever before seeing a 0. So essentially, the machine has learned a mixture model with the 0 and the 1s, and it's very hard to cross the barrier to go from one digit to another digit, well, if you want. So there have been a lot of tricks in statistical physics to try to solve this problem, how to sample faster, okay, how to speed up sampling. So it's a problem that people have been studying for a long time. And one trick, which is very useful, one technique, which was very useful, is called parallel tampering, which was invented by Swenson and Wang in the 80s, which is called also replica exchange Monte Carlo. And the idea is that if you want to sample a Hamiltonian at a given temperature, which is usually low, then you may be trapped locally in some, bottoms of the wells, and you never explore the other wells. So what you can do is, instead of running one simulation at one temperature, you may run many, many simulations at many increasing temperatures, starting from the one you are interested in up to very high temperature. So at very high temperature, the sampling is very fast because the landscape is almost flat. So you can explore a lot. And then if you do coupling between nearest neighbors temperatures, so you can exchange configurations, then you will actually transfer the large exploration, the fast exploration at the high temperature level to the low temperature levels, and it will help you actually crossing barriers and go from one well to the other. Okay, this is very efficient if you have many temperatures which are next to each other. So you need a chain of temperatures going from the one of interest up to very high values with many intermediate points. But in fact, if you think about it, that's a very nice and general principle, but there is no need to think about it only in terms of rescaling the Hamiltonian by some temperature, right? What you need is a Hamiltonian you want to sample, a distribution you want to sample, and then other ones you can sample in an easier way. They don't have to be exactly the same energy over a global factor. There could be any kind of other Hamiltonians which are kind of similar to each other. And the last one being very easy to sample. Okay, so we use this idea to introduce another technique, which is kind of parallel tempering, but specific to restricted Boltzmann machine, which we call stack tempering, which is a very simple idea. If you run your data from one RBM, you are in dimension n, you learn representation in dimension m. Now, you can think of those representation you have learned as kind of new data, metadata for another RBM, right? Another machine, which is going to learn representation of the representations. And it will be in other dimensional space, even smaller, right? And you can use these representations of representation as kind of metadata for another RBM and so on. So what you do when you do that, you learn models which are simpler and simpler, which are more and more constrained from the original data. The last model you learn, which is the smallest RBM on the top of a pile, of a stack, is actually a pretty bad model, right? In a sense, it doesn't represent the data in a nice way. Actually, the size of the visible and hidden configurations the sizes don't even match the one of the data. It's kind of highly compressed and constrained version, right? But it doesn't matter because you can think about them as different points along these chains as in parallel tempering, and they will each run their Monte Carlo chains, and you can exchange configuration from one to the other because the size of the visible layer of a machine here, machine n+1, matches the one of a representation of machine n. So you can exchange them, and the very small and very fast machine on the top will actually help sampling the bottom machine, which is the one of interest, which generates new data. Okay, and I just want to mention that this is very efficient, and in fact, it's more efficient than parallel tempering, because if you do that on actual data, you need much less intermediate temperature points. You need only a few RBMs to do that, and it samples very fast. Okay, so it's an effective way to go in the direction of deep Boltzmann machine, which makes sampling very efficient. Okay, if I still have a little bit of time, yes, I would like to go to another.
Professor Monasson, unfortunately, we have not a lot of time. So if you wish to have some time for questions,
yes, I will show just one slide.
Yes. One slide, it's okay, please.
Okay, so the last thing I wanted to mention is that there are very interesting also questions which are actually still not fully resolved if we look at representations. So depending on the hyperparameters of a machine, the way the data are represented, the same data can be represented in many different ways. Here I'm giving one example which is extracted from this paper, where depending on the size of a hidden layer in these machines, you see that you may be in two completely different representational regimes. One regime where the hidden units are essentially looking at representing kind of prototypes, right? And so that means that each hidden unit is representing some kind of average digit, kind of model digit. And another regime, when if you have many of these hidden units, you can represent local patches, okay? that you need to combine in order to make up a digit. And the right representation with these local pages, it's much more powerful than the one on the left. Because by combining all these local pages in many different ways, you can actually have a combinatorial power of expressing many rich digits, right? You have a rich expressive power. And that's an interesting regime where actually we try to force the machine to learn in order to be efficient and to have a high entropy in terms of generating new data. Okay, so there are a lot of open questions about that. Just want to mention that actually we can also do more than that. We can actually try to bias the way the machine is learning to try to put some information, localize some information on the latent factor to do, for instance, some kind of morphing when you can actually keep a lot of features of your data and try to modify some of them. So for instance, how you move from faces on the left here to the same faces where you have added glasses, but you have not changed anything else. So you can change one binary attribute by sliding real variables here. So that's nice. You can do a lot. And I will end up with the conclusion. So historically, I think Boltzmann machines are very important because they were the first effective generative models that could be learned from data. So we talk a lot nowadays about, DALI, ChatGPT, all these generative models, which are super powerful, but these ones were the first one. They had also a profound impact on neural network research at the beginning of the deep learning era, which I just mentioned briefly. And I think they are still very much interesting in a variety of contexts nowadays. First of all, to investigate some theoretical questions. A lot of things about how this machine learns is not understood from a theoretical point of view. There are still a lot of obscure points, so I wanted just to briefly mention that. So they are interesting. They are also interesting for practical application for limited data sets. There are a lot of applications where the data are not so large. The data set is small. And these machines actually are quite controlled. They have few learning parameters, so they can be used. And I think they are also interesting in the field of neuroscience. There is growing evidence that our brain is able to build internal models of the world in a field called predictive coding in neuroscience. And this machine could be actually some kind of simplified version of these internal models that the brain is comparing to what it sees, the sensory information, and try to adjust these models, right, to make predictions. So I think there are still a lot of bright futures of these machines in the future. Okay, so I will stop here, and I thank you for your attention.
Thank you very much for your talk. And now we have some minutes for questions, please. The colleagues probably in Zoom or in our conference room. Professor Monasson, I just want to ask some question about proteins. You show how this technique can be used to understand how the proteins may be fold. And as I remember, there is a Leventhal paradox related to the folding of proteins. So if you have the sequence of amino acids, and this sequence determines the second order structure of the protein. So this is it should be alpha helix or beta sheet or following organization of protein. So can we use in this case, maybe I'm not right, so can we use this technique as a tool to understand how this protein may be fold in sense of to solve this paradox? So as you know, you need the time about more than the time of universe to take different states, different possible states of proteins.
Yeah. Okay. So in fact, of course, during the folding process, the protein is not exploring all possible conformation and so on. So what is important, in fact, there is some evidence, even with this technique, with the Boltzmann machine, that the folding process, the folding pathway is actually also programmed in the sequence of amino acids just to actually make it faster, right? So there might be some intermediates along the pathway from the denatured configuration up to the native fold, the structure that can be crystallized, that correspond to partial foldings that then are assembled together. Some of them actually are not present Some contacts are not present in the native fold at the end of the folding process. But they have to be there in order to make the kinetics fast. And there is some evidence that some of the effective interactions that can be found with these machines correspond to this kind of intermediate contacts, right? So yes, there is, I mean, not only the native fold, the final structure is actually programmed in the sequence of amino acid, but also the folding pathway. And that can be, say, unveiled to some extent with these techniques. Yeah.
Okay, thank you very much. Now we have problem with electricity, but still we can listen your answer. Thank you for your answer, very interesting. And we have a question from Mark Gorenstein. Professor Gorenstein, please.
Thank you. Thank you for a very interesting talk. My question is the following. Even in the simplest Ising model, we have some smooth behavior of thermodynamic function and also phase transition, critical points, and so on. In this, when we obtain these Ising parameters from some data, it looks that we obtain very, very complicated situation between smooth behavior and the critical behavior of this Ising model. What is the role of this critical phenomena in the whole process? How when we change some parameter, we can go from smooth behavior to critical behavior, and what is this?
Okay, so it's a complicated question. It's a very interesting and complicated question. So I would like to say 2 points. I mean, 2 things about that. First thing is that roughly, if you look at, for instance, this particular application on proteins, there is no evidence that the kind of Hamiltonian you get at the end is in a kind of low temperature phase. It seems, I mean, you could be in a spin glass phase, you could be, you know, it could be extremely complex. This is not the case. It seems essentially the local fields actually, there is a lot of conservation, right? Are strong enough so that actually the landscape, the system seems to be in a landscape, in a part of a landscape, energetic landscape, where actually we are closer to a paramagnetic regime than a spin glass regime, right? So it seems to be quite smooth from this point of view. That doesn't mean there are no local minima, but the system doesn't seem to be trapped forever in some local minima, right? As you would expect in a spin glass regime. Second thing, we have to be extremely careful because some papers reported some spin glass behavior, this kind of thing, but we have to be very careful about the sampling noise. So when we infer a model, we always have a limited number of data available, right? By definition, this is the data set you have. So typically here, when you have proteins which are of, let's say, lengths of tens to hundreds of amino acids, you may have typically thousands of sequences, okay? So now if you have length 100, so the number of parameters you have to infer is of the order of hundreds of millions, because you have 100 square times the number of amino acids squared. So it's a huge number of parameters that you infer from thousands of sequences. So that means that we cannot do that without being careful about the accuracy of what we infer. In fact, this limited number of samples that we have is actually, it could actually create some, we can be very much affected by overfitting, right? So the overfitting is going to match exactly the precise data, and it will create a very rough landscape. So we have to be careful and introduce some regularization to try to smooth a little bit the landscape. So if you do that, you don't get any problem anymore. So I think it's very hard to answer your question because given the limited number of data we have at the disposal, we don't really know where the phase is. But if we smooth it enough to make it reasonable, then it seems to be quite well behaved, and we don't have this kind of low temperature regimes that you would expect in spin glasses, for instance.
Okay, thank you very much. So, dear colleagues, we are not in a program, and we're sorry for some technical issues that we had, but we recorded your lecture, Professor Monasson, and it will be published in our YouTube channel, and everybody will be able to watch this lecture once again. So let's thank once again to Professor.
Thank you so much.
Professor Aurel, please, the floor is yours.
Thank you. It's a great pleasure to start the second half of today's seminar. Our first speaker is Vitalii Tymchyshyn. I hope I pronounce it correctly, from BITP. Will tell us about introduction to neural networks and their physical applications. Please, it's 35 minutes plus 5 minutes questions as for the previous speakers.
Okay, so today I want to make, let's say, a very small talk about the basics of machine learning. And I guess today we will try to build right here, some small neural network. First of all, what I want to start from just a little bit like an introduction. It's the thing that we all know that neural networks are basically flourishing in the recent time. And of course, as far as we know, so they can be used everywhere. So starting from, I don't know, doing something with images or chatbots or whatever we know. But of course, they can be used in physics. And we saw today some applications in physics, and there are a few more let's say, found in different articles on the internet. I'm not a sort of an expert in all of these, so I'm just putting some references down here. So if you are interested in any of those, you can check these references and get the information firsthand. But first of all, they are used as some sort of substitution for the density functional theory, because we know that all these calculations are quite lengthy, and neural networks are a way to get all this information a bit quicker. What else? They are used for computational biology. So we all know that structure of the proteins, it can be obtained with the help of neural networks, and we have recently observed the Nobel Prize for AlphaFold. So it's a really interesting topic. And moreover, they can be used in materials research. So we can try to encode different materials in some sort of ways, I don't know, some strings where we define what kind of what is the atomic composition of the material, so on and so forth. And we are trying to use neural network to predict the properties of this material. And of course, that helps us to screen all over the wide range of materials and to get some idea that this thing may be interesting for us and that is not interesting for us. And this is some sort of potential superconductor, for example, and so on and so forth. What else? Classifying phases of matter, for example, here is one work that talks a little bit about classification of different phases in Ising systems. And here is one more example, a little bit more complicated. This is about so-called toric codes. I'm not sure why it's not seen on the screen. Quite interesting idea by Kyiv, and it seems that neural networks can be used for error correction in these toric codes. So this is quite an interesting topic that involves topology and condensed matters. So we are trying, in some sense, we have some base state of the system with periodic boundary condition. In some sense, we can think about it like living on the torus. And we know that this base state, it is degenerated, and there are different, and we can some sort, let's say, encode topology with the topology. We can encode information on a qubit in this system. One more example is more recent works from, I guess, 2019. It's so-called Cindy autoencoders. This is very interesting topic as well. For example, this one, it takes some sequence, some video sequence, and we show to the neural network some pendulum. So it's just swinging here and there, and we encode with this neural network, we encode the pendulum. And this is so-called autoencoder, so it some sort of compresses all the information we have in video into few parameters in some parameter space. And then we are trying to get from these parameters some sort of equation that will describe our system. And it's very interesting that just showing video to the neural network and getting these internal parameters, we can get with Cindy some sort of equation, so differential equation that describes our system. So maybe in some cases we can use neural networks to predict some physics or some physical laws that we don't know at the moment, but we can gain some information from our experiments and try to use neural network to get something from there. Okay. Now, what about neural network and what it is, how it works? There are a few different ways we can look at the neural network. I call them, one of them I call the engineering way, and the other one I call the mathematician's way to look at the neural network. For engineers, it's just some sort of program that has some free parameters inside. These parameters are calculated with a separate algorithm that we call a training algorithm that gets some data. This is our data set. It gets this data, it does something, then it deduces these parameters. Then these parameters can be plugged into the model we have, and this model is starting to produce something valuable to us. What is more interesting for us is the second view, the mathematician's point of view. I have this image bit enlarged here. So the general idea is the following. So there is a space of all possible functions, everything we can think of. And among these functions, there is the one function that we want to get. So this function does for us something useful. I will show a few examples a little bit later. And we have some hypothesis set that we are working with, and we are navigating through this hypothesis set to get the function which is the closest one to the target function we have. Let's get through this in a few steps. So first of all, let's check out about the function. So we all know that our problems that we pose, they are more or less, they should be somehow converted into this point of view with the function. So for example, here is an example. Suppose this is an image. We can think about the image as a point in a very multi-dimensional space. So it has some pixels. Each pixel has some sort of brightness. So I can make in a many dimensional space. So it's image n by m. So I take r with m by n coordinates, and along each coordinate, I make a step that corresponds to the brightness of this pixel. So at the end of the day, I have one point in this space, and I have a function that transforms this into another point in this space. It can be reinterpreted as an image with the same dimensions. So, for example, if this is, I don't know, a photo of us sitting here, this function can be as well removing the background. We always use this function in Zoom, for example. So if I'm using Zoom, I'm often turning on the removal of the background, and this function can transform the original image into the image without background. One more example, not necessarily we should map between spaces with equal dimensions. I may have many dimensions here, so I'm transforming some image into the point in many dimensional space, but at the end of the day, I want to get some smaller number of coordinates that, for example, represent coordinates of the joints on a hand. So I have a hand, it should, let's say in this image, we see a hand or someone showing his hand, and this function gets coordinates of joints on this hand, and then I can, for example, overlay these joints on the image and show the result. Here is a slide from our previous seminar, and you see that a neural network that was running on my computer has successfully detected a hand and put points where it supposes the joints are. So we basically see that it has worked more or less good. Okay, one more example, a little bit more complicated. So we have an image, and we are trying to get from this image coordinates of the rectangle that should contain a face. Moreover, we are using the second function that takes this face. So we are taking this part of the image. If we need, we scale it somehow, do something with that. And we are transforming into some very abstract space that just has some sort of k coordinates. But what we want, we want this function to be such that if we show images of the same person, we want to get vectors that are close in this space. So, for example, angle between them is close to 0. If we show images of different people, we want to get vectors that are more or less perpendicular. And with this pipeline, we can, for example, detect people and guess who these are. This is once more a slide from our previous seminar, and you see that the neural network running here has recognized at least few of us. So it somehow it worked.
How it knows the names?
I have before the seminar, I have looked at our site in the internet. I took all the photos from there. I passed them through.
You already introduced.
Yeah, I already introduced everyone to the network. So I took our photo from the internet. I passed through this function. So I already knew this sort of vectors for each of us. And then when I showed the image of the auditorium, it was able to recognize at least a few. Okay. So now I have, I guess I have more or less convinced you that most of the problems can be reinterpreted in the way of mapping from one multidimensional space to another multidimensional space. Now, what about machine learning as it is? So any machine learning problem can be described, and any method of the machine learning can be described by answering 3 questions. So first one, how do we choose the family of functions? Of course, there is a caveat to that. This family of functions should be good enough so that we can calculate that on computer. So I can't take, for example, all functions or all smooth functions or something like that. So it should be at least parameterized by a finite set of parameters. So I can't get too many parameters and so on. The second one, how do we measure the goodness of the function? Because in many cases, I won't be able to compare that directly to that ideal function that I have, because I can't calculate that in many cases. So this is the question number 2. And the question number 3 is, how do we maximize the goodness? So, okay, I have some measure of how good my guesstimate is from the hypothesis set. So now, how do I create some iterative process that will get me to the best result I can get? And answering by these questions, we will get some method of machine learning. I will give a particular answer for the neural networks. Well, right now. Okay, first, let's start with some sort of problem. So I want to consider a very simple problem of recognizing some numbers and letters. So we have a data set of numbers and letters. This is an example, some excerpt from our data set. These are images 28 by 28. So it's some sort of extended MNIST, so nothing very fancy. And you may guess that each of these images will be reinterpreted as a point in a space that has 28 by 28 dimensions. And I want to create a function that will map them into, I guess, something like 50 in a vector that contains 50 numbers that corresponds to each possible symbol that I have, each digit, each letter, and so on and so forth. So if I add everything up, it's roughly 50 symbols different. And I want to create such a function that will map them into that space. And each component of the resulting vector will be the probability that this is the symbol that system sees at the moment. So that's what I want to create. Okay. How do I do that? I will create a neural network. So what is a neural network? That's a particular way of defining these sort of functions that we will use for detecting these symbols. So we all know that neural networks, they began as some sort of analogy to the biological neurons. And one way to make this analogy of, or the main idea that people had is to represent the entire function as a collection of small units called neurons. And each neuron, it looks like this. So it takes some inputs, it weights them with some coefficients, W0, W1, W2, so on. And then it uses an activation function on this thing. So we have some linear combination of what entered the neuron, and then we use some function. Which functions do we use? Most often are used, most often we will see one of these functions. So very often revu is used. So I will show it a little bit later in our neural network, or it can be something from the sigmoid, tangent, elu, and so on and so forth. So from these units, I will create, with their combination, I will create my function that I'm interested in. And this function will be, and I will find such coefficients W1 to Wn, so that it will more or less work on the data set I have. Here is an example. Once more, thinking about neurons in model neural networks is a bit hard because they are very, very large. So we are lumping them together into so-called layers. So I have many, many neurons that are collected into one layer, and I'm thinking of them just like a single layer, not as individual neurons in that layer. It's just a little bit easier to think. We use the same thing we used when, for example, I don't know, we think about some electronics or something like that. We don't draw all the individual components or some PN junctions and so on and so forth. So we often draw some blocks, and these sort of these layers are such blocks of neurons. Okay. Here is examples of layers that are often used. For example, fully connected layer. These layers are distinguished by the way that neurons are interconnected inside the layer. So, for example, this one, its characteristic is that all the neurons are getting inputs from all the neurons from the previous layer. So that's why it's called fully connected. It can be convolutional layer. So, for example, when I'm taking some part of the image, I'm multiplying with some coefficients in a matrix, for example, 3 by 3 matrix, and I am adding everything together to get the result that is positioned into the output image. So here is an input that I get to my layer. Here are many, many neurons that are processing this thing. This is one of them shown, and this is the result that I get after processing with this convolutional layer. Very often used. You can even hear about CNN, convolutional neural networks, so it's really heavily used everywhere. Okay, it has some sort of even analogy in the biology, because if we think about how we see the world, it's a very interesting fact that our brain is getting information from the retina, not only from some, I don't know, single rod or cone in the retina, but it gets some information collected from so-called receptive field. And this receptive field, it contains its center that gets directly to the bipolar cell that will send this information to brain. And it gets information from so-called horizontal cell that collects information from this surrounding called receptive field surround. And it turns out that what we see basically in many cases, it's sort of gradients. And it's very similar to the convolutional operation I showed you, because we can get gradients with that sort of convolution by taking some matrices. And this is a very interesting fact, because, for example, that leads us to seeing some sort of interesting effects. For example, if you look at this picture, how do you think which one of these rectangles is brighter and which one is darker?
Below is brighter.
Above is darker. So that's what we think because we see all these gradients. But what if I do so? They are the same.
Yes.
I'm afraid I can't show that on the internet because I can just here hide this part of the picture. But that's actually the effect that is caused by that sort of different fields. And that fields are very similar to the convolutional operation that we use in neural networks. One more layer is a so-called max pooling layer. It takes an image, it splits it into parts, and from each part, the largest number is taken and then sent to the next layer. So it makes this image smaller, but it keeps the biggest values. Okay, with these layers, having all these layers, we can construct a neural network. So here is an example of very simple neural network called LNet. And these are basically layers. So layer 1, here is a stack of layers. They all get information from this layer and then process in each individual way, then more and more and more. And at the end of the day, we get some information like the probability of getting some digit. We will build very similar thing right now. So here are some more networks just to show you different architectures that we have. So there are many different architectures, and depending on how do I compose these layers, I will get different functions and I will get some different architecture of the neural network. Okay, what will we do? Here is example of these layers, but they are expressed in formulae. So that will be a little bit more closer to us. So here is the convolutional layer. So I'm taking the input x. X is some sort of matrix, and I'm weighting these data with some coefficients w, alpha, and beta. Alpha and beta are changing from -1 to 1. So -1, 0, and 1. Here is ReLU, the function I showed you. Its original input multiplied by the Heaviside theta function. So this function is 0 for numbers less than 0 or 1 to numbers that are greater than 0. Max pooling layer. So we are just taking maximums from these numbers. So you can check indices that I'm just considering some small square and I'm taking the maximum. And fully connected layer that I showed you. Once again, I'm just taking everything that we had at the input, so all values in x, and I'm multiplying each one of them with some sort of coefficient wij. So each of these layers has some internal coefficients that are used to train these neural networks. So by changing these coefficients, we are basically navigating throughout the space of hypothesis functions. And if we find good enough coefficients, we will be having neural network doing something interesting for us. And this is the neural network that we will construct. So I will take some input image. I showed you these images above. I will make a convolution. I will make ReLU, one more convolution, one more ReLU, max pooling, and linear. And at the end of the day, I will get vector y that will describe the probabilities of having different numbers or symbols in there. Okay, so here is how this network is constructed in some sort of engineering way. Very similar way. So I'm having this convolution model. I'm creating this neural network right now, and here how it's made. So here is the first convolutional layer. So this one that will be acting on the raw data. I'm setting some parameters. Let's not be so technical about it. Then I'm doing ReLU. That's activation function. Here it is. Then I will perform convolution once again. ReLU, max pooling. This is technical thing. It does nothing basically, and linear. That will give me the result. Okay, so basically this is Python code that constructs the neural network. This is once more its architecture shown from the input and to the output. So convolutional layer and so on and so forth to the output 1 by 47. So I have 47 classes. So not 50, but 47. Okay, so now we have created our neural network. So that's basically we define the hypothesis set of the functions that we will be searching through. How do we define the function is good or not good? You may notice that this thing will give me a vector that contains numbers from minus to plus infinity. What I do, I apply a softmax function. Here it is. You may notice that these exponents, they basically transform that minus to plus infinity to 0 to plus infinity. And this weighting makes all the numbers from 0 to 1. And basically, these are the probabilities of having some class. So I have 47 classes, and when I apply this function to the output of my neural network, that will be the probability of me having certain class. Then what I do? I have so-called data set. So I have images of digits collected by someone, and I know which digits are on each image. So I know so-called true value. So I can calculate this sort of function. This is called categorical cross-entropy loss function. So basically this function estimates how bad my neural network performed. So I feed some data into that neural network. It spits me some result. I get these sigmas, and then I use this function to obtain one single number that tells me how bad the result is. So if this function is, I don't know, high enough, it means that my neural network performed poorly. So bad neural network. If it gives me some low number, we will get some small number, and this smaller number will mean that we have a good result. So that's what we have. And
small
here,
with respect to what?
In, let's say, absolute terms. So I will get some numbers, and I will try to construct an iterative procedure that on each iteration will make this number smaller. So whatever I get, it's good. And here is the technical way to define that. So here, how I define that in Python. So I say I want cross-entropy loss, and I define so-called accuracy function. So here it is. I'm afraid I don't have time to plot this somewhere, but I think everyone gets the idea. What we do next? We perform minimization. So I start with some point. Basically, it's, most often it's some random point. So I choose these coefficients to be random. And then I start some iterations to make this function smaller and smaller and smaller. Which method I have? Most often, so-called first-order methods are used. Here is the whole list of them, so I don't want to get through all of that, but there are many, many methods. Some are good, some are not very good, but anyway, we can use them. You see that the problem is quite challenging, because if we have some complicated function, and we definitely will have that, these methods, they can converge to the point we want, or they may converge to some different points. That basically depends on our luck in some sense. But in many cases, they converge well enough to use that neural network for our purposes. Okay, I don't want to get, how much time do I basically have? 4 or 5 minutes. Okay. Okay, I think I don't have much time to get through all of this. This is just an example of how do we treat neural networks. So I think I just click through very quickly. So first is so-called, so first we construct a neural network. So here are some random functions just to illustrate the idea. So these are neurons. This is our final loss function. In each of these, we have some internal function that takes some inputs and generates some outputs. So these are completely random, some sines, cosines, whatever. What we do, first of all, if we know this function, we know all its partial derivatives. That's very good. Why is good? Because we have a problem of getting the gradient. So we initialize our neural networks. Then we do so-called forward pass. So I take the input data and I pass it through the layers. So in each layer, I can calculate the value of this function. I pass it to the next layer. Here I can calculate the value of this function, so on and so forth. And I can calculate the values of the derivatives. Now I want to get the gradient to make that method work. And how do I do that? I have partial derivatives in each of the neurons, but I want to get the gradient of the function, so which means the derivative of the function with respect to all the parameters that live in this neuron. And I do that with so-called backward pass. So some of them can be plugged in right now. Some of them are calculated. I'm passing derivatives with respect to inputs into these previous neurons, and then I can use them and the chain rule, like df over dy times dy over dv3 is equal to df over dv3 to get these values. So I can calculate them, and so on and so forth. So I'm passing this information and I'm getting all the derivatives. So we can get the derivatives by just simple rules, like I'm doing forward pass by passing inputs, calculating f and partial derivatives and passing the output here. And then backward, I'm getting gradients, I'm multiplying and sending these partial derivatives back. So very simple thing is called backward propagation. If we train our neural network, we will get something like this. This is the loss function, so completely random numbers, but I'm performing these iterations and I'm trying to get this loss smaller, smaller, and smaller. You see two lines. This is train set and this is test set. Train set is basically the set that I'm using to train my neural network. Test set I don't show to my neural network, but I use it as some sort of checking how good my neural network is performing on the data it has never seen. So that's what I want. Okay, I will not retrain it as it takes too long. I just load the pre-trained network. Here is so-called confusion matrix. It shows us how good the network is performing on different data. So here you see all our classes like 0, 1, 2, 3, different letters, so on and so forth. And if this matrix is diagonal, it means that neural network is perfect. So it never misses any class and it works really well. Really fine. Here is how it looks from the inside. Here is an example of an input. So I have an image, 28 by 28, and I'm passing this image through layers. So this is the convolutional layer. It's basically a few layers stacked together, something like 10 of them. And you see the results of performing convolution operation in each of these sublayers. So here they are. Then I'm doing ReLU, this one. Then I'm performing one more convolution, one more ReLU, and the last one, I forgot myself, softmax or something. And then I get the result. This is after applying that sigma function. You see, these are the probabilities. These are the classes. So it's basically that vector y that I showed you in that equations. And you see that number 4 has the highest probability, something like 95% or something like that. So I can use this network to detect that this is number 4. Because
Vitalii,
yeah.
Sorry to interrupt you, but now you should get into the questions. It should be yeah.
Okay. It basically was the last slide that I wanted to show you.
Okay.
Okay, maybe just an illustration. If I send some data that I draw into my neural network, so this is the result of the recognition I get. And basically, that's all that I wanted to show for today. So thank you for your attention. And if you have any questions, please.
Thank you. Thank you. Should we maybe start with questions from...
We have questions in our room, Professor.
Yes, exactly.
Okay,
exactly.
okay.
Let's start there, I think.
Professor Vakhnenko, please.
Sometimes in internet ask us, are you human or not? And your technique allow to recognize non-human as human, yeah?
Yeah, absolutely correct.
So,
moreover, I saw an article when someone basically told how he built a neural network to fight with that sort of captcha on the internet. And some guys commented that it sounds ridiculous because machines are helping humans to prove to other machines that they are humans. So yes, you're absolutely correct. That's really some sort of ridiculous thing, but that's how it works.
Yes. Do we have more questions from the room?
No, we don't have in the room.
Okay.
Maybe in Zoom.
I have a general question. I'm not sure if there's somebody else who wants to raise a question in the Zoom. You have to raise your hand or just shout. We're not that many from, well, we are actually. We are like 20, more than 20. So you can raise your hand if you want to ask a question. Right now, there is no raised hand, I think. So I can ask one question. Would you like to talk a little about, since this is a seminar on theoretical physics, what is the role of theory in this field? How much can you understand and how important is it to understand when a neural network works and when it doesn't work?
Well, if I correctly understand the question, we can use neural networks in many, let's say, branches of physics. And of course,
that's not the question. Of course, we can use it in many cases, but in most cases, at least, it's a trial and error. You use it, and then you see if you get a good result, you're happy. But there is not so often an understanding why it works or doesn't work in a particular case. At least that's my impression from being active on certain aspects of this.
Well, actually, you're quite correct, I think, because we don't have at the moment some really nice theory what happens in the neural networks and why they work the way they work. So I have really seen some articles that, for example, told us something about so-called batch normalization and why we should perform that batch normalization. And then a few years later, I see some different articles that says everything was wrong. They are wrong. We use batch normalization because of something very different. And so I guess we don't really have some nice theory at the moment, and it will be really nice if at some point we will get this theory, I hope at least, and then we will get the idea why some things work and why some things don't work. So at the moment, yes, I really have a feeling that this is a sort of witchcraft. So we are doing something, but we don't have some theory, some solid ground to rely on. So we are just trying something. If it works well, it's great. If it doesn't work, we don't actually know why.
Okay, so then we have the same opinion. I have another take on this is that when it works, it's usually because there is a lot of training examples. You had a training set there. If you have a nice training set, then apparently this witchcraft works. But there are cases when I was actually contacted this morning by two students who wanted to use machine learning to understand the inside of black holes. And I answered, I don't think there are any training examples, so it won't work. Do you think that was a good answer?
As far as I know, recently, many people are using so-called PINs, so physical informed neural networks, as a way to fight the problem of small data set. So if we have a very small data set, we can try in some sort to enrich it with our knowledge about some symmetries of the system or some physical laws that we definitely know that should apply to the system. So probably something can be done, basically introducing some smart loss function that includes laws of physics that should be applicable to the system, and we can get something even from small data sets. Of course,
did you say? What
since we
is it
don't
called?
have some theory, I can't predict will it work or not, but I guess at least there are some chances. So physics.
What was the name? What was the acronym? Physical informed.
PINNS, physics informed neural network. So I don't have chalk here.
Okay.
Right.
Okay. Maybe I should look at this.
Oh, thank you.
Do we have any other questions in Zoom? There doesn't seem to be any. There's no raised hand. Then let's thank the speaker. And let's move on to the next and last speaker. We are, I'm rushing a little bit, but we are slightly behind schedule. So it's Oleksandr Vidybida from, yes, PINS. Okay, I'll have a look. Stimuli classification in a reverberating spike neural network.
Good day, dear colleagues. First of all, I would like to thank to the organizers for launching this scientific event and keeping it alive, and for a possibility to speak here. I will speak about networks composed of biological neurons. Here is a schematic picture of biological neuron. It is a normal living cell which has its body known as soma, dendrites, and axon. Then through the dendrites, the neuron gets stimulation from other neurons. As a result, it can get triggered and generate an output impulse, also known as spike, which propagates through the axon to other neurons. The conduction velocity of a spike lies between 0.5 and 120 meters per second. And the axons can be considerably long. From this nice picture made by the magnetic electronic imaging, we may get an impression what might be the length of axons in a human brain. It is about 10 centimeters or longer. That means that any network composed of biological neurons is a network with delayed interneuronal communication. And this further means that any model aimed at simulating such a network must be aware of its own space and time. So a neuron gets impulses from other neurons, get triggered and send impulses to neurons. But there are also neurons which do not need stimulation from other neurons in order to send output spikes. One example is the sensory systems. These cells are known as, these neurons are known as receptor neurons. In the vision system, it is the well-known photoreceptor neurons. In the hearing, it is the well-known cochlear hair cells, and in the olfaction, these are the well-known olfactory receptor neurons. So these receptor neurons get triggered due to interaction from the external media, generate sequences of spikes to so-called secondary neurons. So a neuron obtains a sequence of spikes. Also, it is often called a spike train. Each impulse obtained by the neuron advances the membrane voltage to some amount, and between the impulses, this voltage relaxes to its resting state. But if a number of impulses are received in the narrow period of time, then the firing threshold can be surpassed, and the neuron generates an output spike. So the neuron obtains the spike trains. What in these spike trains is important from the signaling point of view? There are two different strategies of coding in the neural system. It is so-called rate coding and time coding. In the rate coding, only the total number of spikes obtained in the period t, which does matter. And in the rate coding, in the time coding, the timing of each obtained impulse does matter as well. Actually, these two paradigms cannot be observed in a pure form, but there are some examples. Here is the muscle fiber, and the strength of contraction of muscle fiber depends on the concentration of the acetylcholine, which is released when spikes are discharged into these neuromuscular junctions. And the acetylcholine is consumed relatively slow, so that the precise timing of the obtained spikes does not matter. And here is an example of almost pure time coding. It is the localization of sound direction with the two ears. Here is the right here, the left here, and this secondary, each of these secondary neurons requires two input spikes obtained in the narrow time interval in order to be triggered. So if sound comes from the right ear starts firing before the left one, and the spikes from both ears meet each other in this secondary neuron, which is different as compared is with the sound is just in front of us. This is about coding in the spike trains, but as regards the presentation of real objects from the external world, then in this talk we adhere to the following paradigm. A perceived object is represented in the brain as a specific type of dynamics in a specific neural network. This paradigm is not ultimately proven, but it looks like a plausible one based on available experimental data. Some data we will obtain a few minutes later here. So now we are approaching to our main task. Look at these four pictures. Any physicist or eye physiologist will agree that each of these pictures evokes a completely different activity in our retina. But when it comes to the activity which drives our voice apparatus, then the activity in a magic way becomes identical. Because we will answer a tiger if have been shown any of these four pictures. So what might be a physical mechanism which is able to transform the absolutely different neuronal dynamics at the periphery of the sensory system into the identical dynamics in other places of the neural system? This question has been raised long ago. For example, this interesting paper published by the Institute of Radioelectronics in the year 1959 is very scientifically sound and still actual. Therefore, it is republished by the IEEE in 2007. But let us have a look what more contemporary papers speak about transformation of neural signals. In this paper, the authors observed a kind of unification of signaling obtained as a result of odor simulation in frogs. These authors speak about the temporal sequences of firing in oscillating neural assemblies. They don't speak here about neural networks because maybe because they worked with insects and it is very difficult to follow these very tiny axons in the insect's brain. And this author, based on the experimental results of others, also notice that a kind of unification can be observed during the sound stimuli processing. And in the vision, these authors overtly speak about the dimension reduction framework. And in this work, they have observed that the pattern recognition by the fruit fly is invariant for retinal position. And other papers of this kind also report that the recognition of objects does not depend on their orientation, on their size, on the scene brightness. All these observations gave rise to the following term, condensation of information. The term has been firstly coined in this paper from Biological Cybernetics. The paper is very interesting. It is logically correct and philosophically correct as well. But as far as I was able to understand, they do not propose any physical mechanism which underpins condensation of information. So in order to have some guess about what could be that mechanism, we now make a short psychophysical experiment. In the place of this cross, pictures of different objects will be flashed. Each picture will be shown for time less than 50 milliseconds. Everybody tries to recognize the objects, and I bet that everybody will recognize at least one object. So the time which is required to transform initial visual stimulation into a coherent object is very short. It seems that the corresponding mechanism has nothing to do with the mechanisms known in the so-called wet biology, because most of them are rather slow. It should be a kind of a physical mechanism. So as a possible mechanism, it is proposed a self-organized transformation of the spiking pattern, both in time and space, after it was evoked by a stimulus. The self-organization in the time domain, the idea of self-organization in the time domain was firstly proposed by Donald McKay, an outstanding physicist and cybernetics a long time ago. Here are the figures from his paper, and in the figure 2, it is stressed that the time delay in the propagation of impulses may play an essential role in the processing of information beared by those impulses. And now let us have a look, let us see how it may look like in a reverberating neural network. So, yeah. Я знаю. Так, це вже безобразие. Okay. So this, sorry? These thick blue arrows denote powerful initial impulses just able to trigger these neurons. After this, these impulses are applied only once to each neuron, and then the network the network dynamics runs on its own. And this dynamics either fades or entrains onto a periodic regime. And the periodic regimes may be different depending on the stimulus which was applied. And the stimulus is a set of times where the neurons were initially triggered. And several networks of this type have been studied in details by means of numerical simulation. Here is the first type. Actually, 20 networks of different sizes have been studied. The size is shown here. This is the radius, the distance between the center of this figure to the center of the neuron. You see 0.5 millimeters and less and less. And the different sizes means different delay in the interneuronal communication, because the propagation velocity is kept the same for every network. And these networks were simulated in the computer program, and we looked for different periodic regimes which the network may be entrained on. And it appeared that a network can be entrained onto several periodic regimes, depending on which stimulus has been applied. The more generous is the network number 9, which can be entrained onto 485 different periodic regimes. And that means that this network can classify or can split the total set of stimuli into 485 different domains. And each stimulus is put in the corresponding domain based on which periodic regime it evokes. Here is the number of periodic regimes found for each net. The net number 9 is here. You see about 500 different periodic regimes. Another network studied was in only a single one. The size you may see here, the distance between the nearest neighbors is 1 millimeter. And again, the network is fully connected, and the stimulus is a set of initial triggering times which is 8-dimensional because the first impulse kept always constant. So here is an example of a single stimulus. The point, for example, this point tells us that the neuron number 8 obtained its initial stimulation at the moment 0.2 milliseconds. The total number of stimuli studied is shown here, 390,625. And we have found 102 different periodic regimes in this network. Here you see the sizes of corresponding domains. Here's the size of domain, and here the number of domains of this given size. So these three stimuli are belonging to the same domain. This may resemble our picture with the tigers, because if our network has been shown any of these pictures simply by triggering its neurons in the specified moments of time, then it will entrain onto the same periodic regime, and therefore it will classify all these pictures as having the same type. Here are the characteristics of the periodic regimes. The period is from 3 milliseconds to 10.4 milliseconds, and this is the number of corresponding different periodic regimes. The most often we get period 3.6 milliseconds. And here we see how many times each neuron fires a spike when going through a single period. For shorter periods, it is only one time, and for longest period, it fires 3 times per period. Here is the histogram of the relaxation times. This is the time required for the network to entrain onto the periodic regime after the initial stimulation. We see that these times are concentrated in the range of several tens of milliseconds. Compare this with our psychophysical experiment, where the time necessary to obtain the coherent image from the initial stimulation was as well in the range of several tens of milliseconds. And here we see the routes of trajectories entraining onto periodic regime. They can merge into a single one before they get at the periodic regime, or they can merge at the moment they get onto periodic regime, and also they can merge some time earlier and jump onto the cycle in several different points. These are the routes, only 3 from the many possible. So now we can do our intermediate conclusion. The classification of stimuli in a spiking reverberating network may happen due to self-organization in the space-time, which results in merging trajectories evoked by different stimuli into a single one. But why do they merge? In order to figure out what could be the physical reason for merging the trajectories, we have dumped all trajectories into separate files. Here you see one file of trajectory. Here we have the whole state of our network shown at the 5 consecutive moments of time. And the separate program was used to figure out what happens when trajectories are merged into a single one. It appeared that each merging of two different trajectories into one is accompanied by the firing of one or several neurons. And indeed, the firing may be the reason for merging, but not every firing results in merging. Here we see two different spike trains applied to a single neuron, to the same neuron. The difference is only in the arrival time of spike number 4, here and here. But it so happened that the triggering spike is obtained at the same time. As a result, in both cases, a spike is generated at the same moment of time, and the difference in the arrival of in the T4 has been forgotten. Here you can see the same effect on another neuronal model. This model requires 4 spikes in its internal memory in order to be triggered. We see that in 3 cases, the neuron is stimulated by different spike trains, but the triggering spike is obtained at the same moment of time, and therefore the output is identical, the spike obtained at the same moment of time. And now a few words about the signaling function of a generic neuron. In his book, The Astonishing Hypothesis, Francis Crick says that the main function of a neuron is to receive signals and to send them out. And by developing this statement, it is possible to propose the following signaling function of a generic neuron. This input impulses, which a neuron can feel. I recall that the neuron is the living cell, and it is an independent living object. So it can feel these input impulses, and if this we call this an elementary events. If these elementary events are obtained in a narrow time interval, the neuron interprets them as being emanated from a single coherent compound event which happens somewhere here in the external world. It do this by means of triggering and sending an output spike. The output spike can be considered as the abstract representation of that expected coherent compound event. And this spike, of course, does not keep information about the timing of each this elementary event, but this is normal for an abstract representation. So it seems that I have no time to speak about the inhibition. Those interested are invited to look through this article in the Wikipedia and some concluding remarks. So we have modeled, we have simulated some reverberating spike in neural networks with delayed interneuronal communication. And of course, they are rather different than the real biophysical neural networks. Here are shown some differences, but we still claim that these simulated neural networks are able to classify input stimuli into different classes. And it is just what it supports the real biological neural networks do. But is it really difficult to break out a set on different subsets? Anyone can do the job, but by applying some reasonable rule like this one. On the other hand, let us have a look on the breaking up the set of initial stimuli for this network. This is the cross-section of the four-dimensional space of initial stimuli with the plane. Here is shown which plane, and to the right a little bit another plane. Looking on this peculiar and complicated separation of the stimulus set, I might venture to say that this little uneducated, funny network, if obtained a suitable learning mechanism, could be trained to do some practically useful job. And finally, I would like to make a comparison of the technical network we have heard about in the first three talks here with real biological networks. Actually, they have more in common than they differ, because both are made from neurons, which are the threshold units. Both have tight interneuronal communication, and both have a tunable connection strength of this interneuronal communication. As regards physical difference, it is not a problem because both networks are aimed at the processing of information. I could draw an analogy with a radio receiver. It is known that a radio receiver may be composed of electronic tubes or semiconductor transistors. The physics is rather different, but both are able to receive the broadcasting from the space. But there is one difference which should be noticed. It is a scale difference in the number of tunable elements. The contemporary, the most modern large language model, it is advertised as having 175 billions of tunable elements. And the number of tunable in the biological network, each interneuronal connection known as a synapse is the tunable one. It is possible, based on the available experimental data, to figure out how many pyramidal neurons are in the human brain, how many synapses it has, actually 30,000 and more, to multiply and obtain this number. So we see that in the brain, 4 orders of magnitude more tunable elements. Of course, this is for today, and the situation can be changed in the future. Thank you for your attention.
Thank you a lot. We are running a little bit over time. I would say let's limit ourselves to just a few questions. Already some people had to leave. I could see that. Let's start see if there are questions in the seminar room in Kyiv.
At the moment, we don't have questions. Probably later.
Yeah. Yes. Is there somebody else in the chat, or who would like to put a question in the chat, or somebody else in the Zoom who has a question? In that case, either raise your hand or just unmute yourself and speak your question. We are only 23 participants, so that is 21 excluding 20 excluding me and the lecture room and the speaker. Is there any question among the Zoom participants?
And blessing.
Yes,
Eric.
Yes.
This little experiment that was conducted, we were supposed to recognize at least 2 images. Was that my understanding? I think I did recognize two flowers and a bicycle.
I saw a bus. Yes, there was a bus.
Yeah.
We can try once.
But it was much more difficult to remember. One had the feeling
Yes.
one recognized everything, but then it's forgotten when the experiment is
Yes.
over. Is that the correct?
Sorry?
My impression was that I recognized everything, but after the show, I had forgotten most of them.
Yes, that was my
It makes it
impression.
happen. You see, actually, the transmission lines in the internet may somehow change the timing. I'm
Okay.
afraid.
Yeah.
Therefore, it may happen.
Yeah. Yeah, there was a bus, wasn't there? There was a bus, I think.
Там був автобус.
What?
Автобус був.
What? Yes, of course. There was bus, there was bicycle, and something else.
Yeah.
Flowers. Yeah.
Look,
Yeah, it was interesting
it
experiment.
was very nice. I was also quite impressed by this experiment.
Yes.
And it's a very nice demonstration that you recognize things very quick time scale. It's very nice. It was very nice. Should we maybe end there? We are running 10 minutes or 11 minutes past the hour, and I suppose that we will have a next meeting sometime next year, but that can be decided offline.